Hi students, this is A. Mandri Kumar Rajesh. In today's video, I would like to teach you cross called dihybrid cross, and also I am going to explain the law of independent assortment with the help of dihybrid cross. Let's start with dihybrid cross. First, I give you the definition for dihybrid cross, then I will explain the definition with the help of a cross. So, first I give you the definition. Dihybrid cross is a cross between two individuals which is made to study the inheritance of two pairs of alleles belonging to two different genes. So it is a cross between two individuals which is made to study the inheritance of two pairs of alleles belonging to two different genes. Before we get into my explanation, I would like to tell the selected traits for dihybrid cross by Mendel. Mendel have chosen two characters for dihybrid cross. Mendel have chosen two characters for dihybrid cross. The first one is seed shape and the second one is seed color. In seed shape, we have two possibilities of traits. They are round and wrinkled. In the case of seed color, we have two possibilities of traits. They are yellow and green. In seed shape, the round trait is dominant over the wrinkled. In seed shape, the round trait is dominant over the wrinkled. In seed color, the yellow color is dominant over the green color. Then I am going to write the allele for round trait. So the allele for round trait is capital R. The allele for wrinkled trait is small r. And for the seed color, in yellow, the allele for yellow color is capital Y. Then the allele for the green color is small y. So Mendel have chosen two characters for dihybrid cross. They are seed shape and seed color. In seed color, we have two possibilities of traits. They are round and wrinkled, and they are represented by the alleles capital R and small r respectively. In seed color, also we have two possibilities of traits. They are yellow and green, and they are represented by alleles capital Y and small y. Now I am going to figure out the dihybrid cross. For any hybridization experiment, we have to consider three major steps. They are selection of true breeding parents and cross pollination, and the third rule is self pollination. So we have to select true breeding parents. Now I am going to write the parental phenotype of homozygous dominant parent. So round yellow is the parental phenotype. This is the homozygous dominant parent. Cross with homozygous recessive parent. So the phenotype is wrinkled green. So this parent is a homozygous recessive parent. The round yellow act as female. The wrinkled green act as male. Now I am going to write the genotype of the round yellow. That is capital R, capital R, capital Y, capital Y. So the capital R represents the round trait. The capital Y represents the yellow character. Here also I am going to write the genotype of wrinkled green. That is small r, small r, small y, small y. Now I am going to explain the definition of dihybrid cross with this step. I told you it is a cross between two individuals. It is a cross between two individuals which is made to study the inheritance of two pair of alleles. Two pair of alleles belonging to two different genes. So this is the first gene, this is the second gene. So it is a cross between two individuals which is made to study the inheritance of two pairs of alleles belonging to two different genes. This is called the dihybrid cross. Now I am going to continue the cross. So capital R, capital R, capital Y, capital Y, small r, small r, small y, small y. So I am going to make a cross between these two individuals. So gametes should be formed by the process meiosis. So the gametes are produced would be capital R, capital Y by the process meiosis. Here small r, small y produced by the process meiosis. Then according to the second stage of hybridization experiment, we have to make a cross pollination. We have to make a cross pollination. So we are going to make a cross pollination. As a result, we get capital R, capital Y, small y. It shows the phenotype round yellow, and they are said to be hybrids, So in F1 generation, we get all the young ones are contains dominant character, that is round yellow. Then according to the third stage of hybridization experiment, we have to self-pollinate the F1 generation. We have to allow the F1 generation to self-pollinate. So we have to cross F1 with F1. That means capital R, capital Y, small y crosses with capital R, capital Y, small y. Now I am going to teach you how to take gametes for these two individuals. So please listen carefully, because most of the students felt uncomfortable while taking the gametes for the individuals. So please listen carefully, my dear boys and girls. Let's go to the board. First take capital R and it combines with the capital Y. This is the first gamete. Then the capital R going to combines with the small y. This is the second gamete. Then the small r is going to combine with the capital Y. This is the third gamete, and the small r going to combine with the small y. This is the fourth gamete. This is how we have to take gametes for the individual. Cross with the same condition will occur here. So capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So this is how we have to take gametes for the individuals. Now this gamete should be plotted in the Punnett square. So now I am going to draw the Punnett square. You have to draw 4 rows and 4 columns for dihybrid cross. In rows you have to place the male gametes. In columns you have to place the female gametes. So capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Likewise in the female gametes also, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now I am going to make fusion between these gametes. So please listen carefully. First I am going to make a fusion between these two gametes. Capital R, capital R, capital Y results in the young one, and it shows the phenotype round yellow because it contains all the dominant alleles. Then capital R, capital Y fused with capital R, small y. Capital R fused with capital R, capital Y fused with capital R, small y. So capital R, capital Y, small y, and results in round yellow. Then the capital R, capital Y is going to fuse with the gamete small r, capital Y. Then capital R, small r, capital Y, and it gives the young one round yellow. Then capital R, capital Y is going to fuse with small r, small y. So capital R, capital Y, small y, and it gives the young one round yellow. Likewise you have to make fusion of gametes. So capital R, capital Y, small y, it shows the phenotype round yellow. Capital R, small y. Here we have to carefully write the phenotype of the young one, because capital R, small y. That means it shows the phenotype round green, because here we have recessive alleles, so it shows round green. Then capital R, capital Y, small y. Then it shows round yellow. Then capital R, small y, small y. Then it gives round green. Then small r, capital Y going to fuse with capital R, capital Y. So capital R, small r, capital Y, it shows round yellow. Then capital R, capital Y, small y, and it shows round yellow. Small r, capital Y. Here we have the seed shape is recessive. That means wrinkled yellow. It shows the phenotypic character wrinkled yellow. Then small r, capital Y, small y. That means it shows wrinkled green. wrinkled yellow, because it contains the capital Y allele. So it shows wrinkled yellow. Then capital R, small r, capital Y, small y. Then it shows round yellow, the phenotypic character round yellow. Then capital R, small y, small y. Then it shows round green. Then small r, capital Y, small y, that means wrinkled yellow. Then small r, capital small y, then it shows the phenotypic character wrinkled green. So this is how we have to fuse the gametes of two individuals. Before count the proportion of young ones, I would like to tell you the phenotypes which are formed from this F2 generation. I want to tell you the phenotypes which are formed from F2 generation. So in F2 generation we could observe 4 types of phenotypes. They are round yellow, then round green, then wrinkled yellow, then the final one is wrinkled green. So we can observe the 4 types of phenotypes in F2 generation. Then look at these 2 young ones, these 2 phenotypes from the F2 generation. It resembles the parental phenotype. It resembles the parental phenotype, that is round yellow and wrinkled green resembles the parental phenotypes. So they are said to be parental type. That is round yellow and wrinkled green are said to be parental type. Then you could observe the round green and wrinkled yellow. It does not resembles the phenotype of parents, and they are said to be recombinants, So the round yellow and wrinkled greens are said to be parental type. Round green and wrinkled yellow are said to be the recombinants. Now we are going to count the proportion of the phenotypes or the young ones. First, I am going to count the proportion of round yellow. 1, 2, 3, 4, 5, then 6, 7, 8, then 9. So we could observe 9 round yellow. So the proportion is 9. Then for round green, 1, 2, then 3. So for round green, the proportion is 3. For wrinkled yellow, 1, 2, and 3. For wrinkled yellow, the proportion is 3. For wrinkled green, the only one young one. So the proportion is 1. So the phenotypic ratio of the dihybrid cross is 9:3:3:1. The phenotypic ratio of the dihybrid cross is 9:3:3:1. So this is how we have to did cross for dihybrid cross. Then I am going to explain the law of independent assortment, which was proposed by the Mendel. Let's start with the definition of law of independent assortment. when two traits are combined in a hybrid, segregation of one pair of characters, is independent to other pair of characters. This is called law of independent assortment. When two traits are combined in a hybrid, segregation of one pair of characters is independent to other pair of characters. This is called law of independent assortment. Now I am going to tell you how Mendel constructed the law of independent assortment. Mendel considered each characters independently. For a dihybrid cross, Mendel have chosen two characters. They are seed shape and sheet color. In seed shape, we have two possibilities of traits. They are round and wrinkled. So round trait is dominant over the wrinkled trait. Here also we have two possibilities of traits. They are yellow and green. Here yellow color is dominant over the green color. So I told you earlier, Mendel considered each characters independently. Now I am going to count proportion of the round trait. So the phenotypic ratio of dihybrid cross is 9:3:3:1. So now I am going to count the proportion of round trait. So here we have 9 round and here we have 3 round. So 9 plus 3, 12 round traits. And in total we could obtain 16 young ones. That is 9 plus 3, 12, 12 plus 3, 15, 15 plus 1, 16. So among the 16 young ones, the round traits appears in 12. So 12 by 16. So 12 by 16 can be written as 3 by 4. And we can write 3. Likewise, we are going to count the proportion of wrinkled trait. Here we have 3 and here we have 1. So in total 4 by 16. So we can return this 1 by 4 and we can write 1. So the ratio is 3:1. Here also we are going to count the proportion of yellow trait. So first yellow, 9 plus 3, total 12. Total young ones are 16. So 12 by 16. So we can convert it into 3 by 4. Then green trait, 3 plus 1, 4. So So 4 by 16 we can return as 1 by 4. Then 3:1. So please look at these two ratio. So these two ratios are similar to the monohybrid ratio. It shows the segregation pattern of monohybrid cross or dihybrid cross or trihybrid cross is universal, that is same in all crosses, and the gametes are the pair of characters are independent from the other character. One pair of characters are independent from other characters. By that only they can assort independently. This is how Mendel constructed the law of independent assortment. With this information, I am going to conclude this video session. I hope the information will be useful for you. If you have any doubt, please clarify with me. Please stay home and stay safe. Thank you very much for watching this video. Thank you.